Okay, so the importance of protein synthesis is that not only does it make you, but if DNA changes happen while the DNA is being copied, these cause mutations, and the mutations cause differences in protein synthesis, which causes differences in life. So mutations happen on two different levels, chromosomal and gene. And so first we're going to take a look at the chromosomal mutations, and then we'll look at the genes. There's 5 types of chromosome mutations, and so these would be deletion, inversion, translocation, non-disjunction, and duplication. A deletion of a chromosome is where a section of a chromosome is deleted. So if we look, this middle peach-colored section is deleted, which makes our chromosome shorter. So if the chromosome originally had 100 genes on it, and then 20 of them got deleted, then we would have an 80-gene chromosome. So it would be shorter because the section would have been deleted, meaning that the chromosome now has less genes. A chromosome experiences inversion where a section is flipped upside down. So if we have a sequence of DNA right here, and the sequence is flipped going in backwards order. So here's an example. If we had like GATC, let's say the sequence is flipped, so then it goes CTAG. If the sequence is flipped, that is because of inversion in the chromosome. So the sequence would be flipped and then would reattach into its certain spot on the chromosome. So the length would be the same, but the sequence or a section of the sequence in the middle would be reversed. A chromosome will go through duplication if a section of a chromosome or a sequence of genes is copied twice. And so that doesn't mean the entire chromosome is doubled in length, but only a section of it. So let's say this chromosome is 100 genes long, and this section is 20 genes. Well, that 20 gene section gets duplicated, so this would be about 40 genes. Since we have added on another 20 genes, the new chromosome would be about 120 genes long. Translocation occurs between non-homologous chromosomes. Now, these are different colors, which show that they're from different parents, but non-homologous means that they're not the same size, meaning they're not the same chromosome. So even though they come from different parents, we're talking about a large chromosome versus a small chromosome. So different colors, but also different sizes. Different color, different size. And so it's basically the same as crossing over, except you're doing it between non-homologous chromosomes. So that section from chromosome 20, from dad's chromosome 20, if we say this is dad, this is mom, will be attached to mom's chromosome 4. And then mom's chromosome 4 attachment will be added on to dad's chromosome 20. Non-disjunction, this is what causes Down syndrome. And so non-disjunction is where we have a pair of sister chromatids not get pulled over to the correct cell during anaphase. And so this guy ended up getting pulled over to the what you would see as the top side. And so when the cell divides, we have two gametes that have an extra copy of this chromosome. And so when you have an extra copy of the chromosome, that is the result of non-disjunction. So if we're talking about a specific chromosome such as chromosome 21 in humans, this is what causes Down syndrome. So in what's called trisomy 21, which meaning 3 copies of chromosome 21, sperm, for example, could have 2 copies of chromosome 21 instead of just 1. And then when it meets up with the egg cell that has another copy of chromosome 21, you get 3. And so here's kind of a map of our chromosome mutations. So here's your original DNA. If we look at duplication, gene D has been duplicated, so there are 2 copies of D. If we look at deletion, D is gone. If we look at inversion, the whole DEF sequence has been flipped. And then we have another example of inversion where the BC segment has been flipped as well. So those are two examples of inversion. We've covered chromosome mutations, so now let's move on to gene mutations. There's three types, and they're all called point mutations. Substitutions would be our first type that we'll cover. And then there's also something called frame shift mutations, and these are insertions or deletions. So first, let's cover substitutions. Substitution is actually what causes sickle cell anemia. So here we have a picture of a sickle cell, a sickle red blood cell, and that's caused by substitution of one nucleotide in your DNA. And so if we have our DNA sequence, we have what looks like CAG, 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 and that lines up with our original DNA sequence. Well, then we decide to change that C to a T when the DNA is getting copied. If we replace the C with a T, the sequence is the same length. We have switched out one of the letters or one of the nucleotides. Well, switching that C to a T has caused the amino acid for that codon to be a stop codon, which means our polypeptide chain right here is shortened. Now, last but not least, we have the two types of frame shift mutations. And so frame shift mutations are where if you add or delete a nucleotide instead of substituting it, you can change the entire strand. So here's the thing. If we have a sentence that said the fat cat ate the wee rat, and we change we take the T in cat and we delete it, then that causes the A to shift over and the T and the E, and this whole section has to shift over one letter. But the spaces stay the same. So what that means is we now have 1, 2, 3, 4, 5 different words than we did before, and one of them is only 2 letters long. And so this is how deletions work, and here's an actual example. If we look at a deletion, if we go down to this side, we have cat, 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 and then we delete the A in our fourth cat codon. This causes the T to shift over, and so it now says CTC instead of CAT. The next one will say ATC, and so will the next one and the next one. But instead of cat, 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 you have different codons, and thus you have different amino acids, making a different polypeptide chain. Whereas if we insert an extra nucleotide, so substitution is where the sequence stays the same length, but you switch one of the letters with a different letter. This is where you don't switch anything, you just add a new letter. So we have cat, 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 and then we add an A between this cat and this cat. Well, this is also going to change our codon sequence, and so we have ACA instead of CAT. Then we have TCA, and then TCA, and then TCA. So like in a deletion, this has changed our proteins, or amino acids. So we have new amino acids here, and thus we have a different polypeptide chain, just like with the deletion. So the punchline is you need to know that 2 different levels that the mutations happen, chromosome and gene. You need to know the types that we've covered in these notes, and you need to understand that gene mutations can change the amino acid sequence, which changes your protein structure or your polypeptides. And mutations are what make us different.